For the first time in history, genetic therapy-based vaccines have been produced at very large scale. Genetic vaccines contain a carrier designed to transport the vaccine genetic material called the cargo inside the patient cells. In DNA-based vaccine, the RNA coding for SARS CoV-2 spike protein is identified and extracted by advanced bioengineering techniques. Cell penetration is complex and requires a carrier to push the cargo inside the vaccinated patient cell. Current approved DNA vaccine include the Johnson & Johnson vaccine and the AstraZeneca vaccine, which use different carrier molecules to push the cargo inside the cell nucleus. Vaccine active proteins are collected and concentrated by centrifugation. Then, vaccine proteins are purified by ultrafiltration or colon chromatography. Vaccines contain excipients, among them adjuvants such as aluminum salts, preservatives to prevent the vaccine from being spoiled, such as ethanol, stabilizers to make sure the vaccine is able to stay effective for a long period of time, surfactants, and for most vaccine, water dilutes the vaccine ingredients. Upon intramuscular injection, the vaccine molecules are progressively removed from the injection site by blood and lymphatic circulation. To induce an adaptative immune response, the vaccine main target is antigen-presenting cells such as dendritic cells. After internalization, DNA vaccine will dock on nuclear pores complex and deliver its DNA into the patient cell nucleus. Vaccine DNA will be integrated in the patient genome and transcribed into mRNA in order to get out the nucleus. Ribosomes in the endoplasmic reticulum will translate vaccine mRNA into a modified SARS CoV-2 protein. Among the possible shaping routes, spike protein might be degraded into fragments by lysosome enzymes. First, spike protein is broken down into peptide fragments through the action of protease. MHC2 molecules then bound to spike protein fragments and move to the cell membrane to present spike protein fragments to adaptative immune cells. Following vaccine antigen presentation, naive T cells will be activated and differentiate into helper T cell and killer T cells. In their turn, helper T cell will activate B cell to generate memory B cell and plasma cells. Plasma cells are antibody producing cells against vaccine antigen, i.e. against SARS CoV-2 spike protein.